Swayam Prabha. Digital India. Educated India.
Dear participants and students, in this lecture, we'll discuss about the bioprinting of kidney. So as I've discussed in case of bioprinting of heart in the earlier lecture, so we start with the motivation for doing a bioprinting of kidney structure, and also then we'll discuss some examples of 3D printing of different kidney tissue structures like the till now the printing of whole functional kidney is not yet achieved because there are various technological challenges in that. So we'll discuss some of this thing in this particular lecture. Let us start with the motivation why kidney bioprinting can be a major breakthrough. Like kidney failure is a global challenge. If you see, there are many patients who are uh, who has this end stage kidney failure where the kidney has to be transplanted, and there are actually many patients they are waiting for say the kidney transplants, because if you see the number of supply is much lesser than the demand. So there are many patients who are awaiting for kidney transplantation, and for this kind of kidney failure patients only therapeutic option is the dialysis and renal transplantation. So many people they undergo dialysis when they are at the early stage, when by dialysis, by removing the excretory material, the toxins and other materials that the kidney generally that filters out, that the dialysis is a method by which that can be done using dialytic apparatus. But in certain cases when they further advance to the end stage, their renal transplantation is the only option. And there are limited organ donors, and also certain cases risk of tissue rejection, cost, and other complications, those remain as major challenges for renal transplantation also, because there are very less donors available, and also not we need to see the matching of this tissue histocompatibility factor is required, like where the matching is required for transplantation, and also this procedure and the thing is very expensive, and there are many other complications associated with this renal transplantation. So renal transplantation is not very affordable, and also not there are not many transplants available also. So many patients are because they sometimes they die because of waiting for a transplant kidney. So there are these are challenges, and due to this, artificial kidney could be a promising solution for this kind of end stage kidney renal failure patients. But if we try to bioprint a kidney, we need to understand the structural and functional requirement to do that. Actually, if you see, kidney is one of the most complex organs in our body. It has different types of structures, like starting from nano to macro at different levels. The structures are present at different levels, and the function of the kidney depends on all these different structures. And their development, they have also unique developmental cues, and then microscopic spatial resolution. Another important thing to consider here is the kidney. It contains more than 30 different cell types, and their functions are very different. So, creating the structure with these many types of cells is a humongous challenge. And also, the kidney is intricately patterned. The epithelial tubes, named as nephrons. Kidney has this intricately patterned epithelial tubes that named nephrons that carries the filtered thing to the to the what is this called? Dialysis, so the bladder, to the kidney blood, to the bladder. So this tubes, this nephron has different functions. It also helps in first thing the first part of the kidney is that is present. That's a main part where the filtration unit that is called Bowman's capsule. It is that surrounds the glomerulus where actually the filtration of the blood happens, and then from there it the blood carries to the renal tubule, the blood, sorry, the filtered solids that carries to the urine part that goes to the renal tubule. From the in the renal tubule that has different types of tissue where reabsorption, again secretion, all these things happens, and finally it goes to the collecting tubule. From there it goes to the bladder, urinary bladder. So this the different structures are of different scale. They are present in different scales. Okay, so nephron is the functional unit of kidney where there are there are billions of nephrons present in a kidney, and then they do this filtration and finally collecting the urine, and finally sending the urine to the urinary bladder for further release. So this the kidney, so nephron is so recreating the nephron structure is very important for to do this job. And as I mentioned, the first structure present in the nephron is the glomerulus, then there are ascending loop, descending loop, Henle's loop, and all these things are all these structures are present. Finally, the collecting tubule and other structures present. So each segment of this kidney is responsible for its own physiological roles, and then thus a different microanatomical features present in that one structure. If you see in the kidney, different structure has their own anatomical representation. They present at different scales, and their physiology is very different. So that is that we have to reproduce those structures for bioprinting of kidney. Without that, the kidney function will not be achieved. But complexity of this kidney, the kidney, as I mentioned, the kidney structure is so complex and it has so many different types of structures. So this kind of complexity cannot be reproduced by any tissue engineering methodologies, any traditional tissue engineering methodology like what we other do by casting, by this thing. It cannot be achieved. And because each element has to be engineered separately based upon the structure, the microstructure present there, based upon their the particular what are the cell types present there, the particular depending on the typical cell types present in that or in that element. Those things we have to consider while going for biofabric or going for bioprinting. So bioprinting is a technique by which it has shown some possibility of replicating a few of the renal structures like some nephron structures like a part of the renal tubule, a proximal tubule. All these things are printed, and all these things are demonstrated, and physiological functions and have again has been seen in that kind of tissue structures. So bioprinting has got huge prospect to generate bioprinting of full kidney, but still we have not reached that stage. So in this lecture I will discuss few of the 3D models, 3D bioprinted models that researchers have developed over time, and these are very important breakthroughs where researchers have shown some a few parts of this particular organs. Like this first in this first research paper published by Lin et al. In PNS, when they have actually used 3D printing technique for development of complex 3D models of kidney tissue, where actually they have printed a human renal proximal tubule, PT, proximal tubule, renal proximal tubule with convoluted tubular architecture and open lumen structure was printed with extracellular matrix, and then they have formed into a in a microfluidic chip. So they have printed this proximal tubule. This is a part of the nephron PT, and it does several other functions. So their interest is to print, reproduce that structure, proximal tubular structure. And please understand this. This is a convoluted tubular architecture. It has a particular architecture, and also I'll show some of the images from this particular study in the next slide, where you can so and then the lumen is also open in that bioprinted structure. So that is very important. So because if the structure is not lumen, then open lumen is open, then it can serve the function of this transport. Now this tube, after they printed this tube was actively perfused with proximal tubular epithelial cells, because epithelial cells, they create the lining of this proximal tubule where the epithelial cells are there. That filtered thing, and then when it is goes to this, so that part, that luminous structure where epithelial cells are present, result is that. And when they actually perfuse the epithelial cells into that proximal tubule from the luminous side, it got attached to that luminous side, but it enhanced the epithelial morphology and function properties. So that if we don't have a epithelial lining in the proximal tubule, then it cannot function, it cannot recreate the function that is done by the proximal tubule in native tissue. So that's why the a complete epithelial morphology is very much important to generate, and that they have achieved. Then also they have seen whether this kind of proximal 3D binded proximal tubule, whether it can uh, do physiological functions. Like one such thing is, suppose if we inject some nephrotoxin drug like cyclosporin A, if it is injected the body and if it goes to the kidney, it is a highly nephrotoxic drug. So it will destroy the nephron structures, this thing. So epithelial, but so that is like, so to mimic that kind of physiological response, when they injected this cyclosporid A within the system, what they found, that epithelial barrier, as I mentioned, that epithelial cells, when they are injected in the proximal tubule, they create a epithelial lining. So this epithelial cell to cell connection is very important. They create some tight junctions. Because of that, they maintain a barrier, so that the whatever the filtered urine that is coming through the passing through the tubule, that should not get again, the whole thing should not get absorbed. There should be a selective absorption and selective secretion. So that kind of things should go on within the proximal tubule. So the nephrotoxic, what it does when it is injected into their 3D printed TV, that structure, if the epithelial barrier is disrupted, and that is also a dose in a dose dependent manner. So that means at very low dose, that probably that epithelial barrier is not disrupted completely, but when the dose was increased, that epithelial was slowly, the epithelial barrier was disrupted, and that kind of disrupted epithelial barrier is cannot do the physiological function. So this is actually a physiological response can be was seen in a 3D printed model. So that's a very important outcome from this study. The other thing they have also done, what they have also printed another tubule structure where endothelium cells are present. So basically they want to create that tubular vascular structure that is present in case of in the renal in this proximal tubule structure, where a in a tube, two adjacent tubes were printed. In one tube, epithelium cells were perfused, and the other one, endothelium cells were perfused. But these two tubes were present adjacently with a closed loop perfusion, so that the things from one tube to another tube can be diffused by this thing, because they want to try create this kind of that active reabsorption via the tubular vascular exchange of solids, right? Because what happened after filtration through after filtration from the glomerular, whatever the rest salts and solids present in the filtered solution, that will again reabsorbed in case of in proximal tubule. So and also glucose also get reabsorbed from the proximal tubule. So they want to understand whether these kind of physiological functions can happen in a 3D printed models or not. So that's why they have created this a tubular vascular model where, and then they were saying whether this albumin uptake or glucose reabsorption can happen or not. So they have found that this model, the 3D printed model, actually can perform this kind of, can actually yeah, this kind of physiological function can actually happen in a 3D printed model. So that is very important outcome from this study. Another thing also they have seen, suppose in case of native tissue, if some people are when the people generally, when the glucose level is high in their body, that means in case of hyperglycemia, the kidney functions can be highly disrupted. That means, so there what they found, in case of also hyperglycemia, a human-like disease state is formed, which can be reduced by administering glucose transport inhibitor. But again, suppose in case of hyperglycemia, a human disease like kidney, in case of kidney also, that is in case of diabetic kidney. So where if the suppose people have this diabetes where the sugar level is high in their body, that cause a disease to the kidney. So that kind of similar disease can be developed by inducing that 3D printed proximal tubule with hyperglycemia, with high amount of glucose. But in this case, again, this can be reversed by administering glucose transport inhibitor. So that is because typically for this kind of condition, glucose transport inhibitors are generally prescribed, and then they have found also the similar response can be seen by using a glucose transport inhibitor in a 3D printed model. So this importance of this study is here they have shown with 3D bioprinting we can actually develop a convoluted tubular architecture that is for the to develop a renal proximal tubule, and then a physiological responses can be physiological functions can be re-established within this 3D printed model. This thing. So this is an important study here. The for the images from this study where you see this is their concept where they have actually used a this kind of concept where they printed a tubule like convoluted tubule like structures with the help of with this thing with the 3D print here, and then they printed this. Then they what they have done, they recreated this kind of morphology, a tube like morphology where two tubes were printed first with the sacrificial plurionic inks. Plurionic ink was used to create this kind of this sacrificial template, tubule like template, and also this the ECM material was used to print the structure, right? So then when this structure is printed, then this ECM was again put on top this, and that was cast, and then the plurionic is actually the advantage is if you reduce the temperature, the plurionic is can be plurionics can be easily taken out. So then it leaves a lumen like structure, a channel like structures within this thing, and there then in one channel they have perfused epithelial cells, and on the other channel they have perfused these endothelial cells. Then they create these two other things. So here if you can clearly see approximately vasculature, a simple structure and a convoluted structures can also be printed with this thing, and then these two channels are connected to, they are very connected to, they are very much connected, and you can clearly see the PT proximal tibial and vasculature where PT, in case of PT the epithelial cells are there, vasculature the endothelial cells have been perfused, and then you see the structure that is present here. This is schematic, and then the simple and this thing is present, and whole mount immunofluorescence staining was done, of the 3D is done, and this is that, and then can clearly see here the sodium potassium ATPase pump that is actually actively involved for pumping out the some of the means that helps in some reabsorption of this thing. So that is there. CD31 is the for the endothelial cells, and nucleus is the same. So this is the endothelial channels for the vascular channels, and this is the proximal tubule channels. So all these two cells are sitting next to each other. So that's thing, and then this is the further magnification of this thing. So this is their channel they have presented, and here this the difference between separation between the PT and vascular conditions are only 70 microns. So that much fine closed loop they have recreated, and it shows also the high magnification of the structure. So this study is as I earlier mentioned, they have also seen the different physiological functions here. Please go through the literature for further understanding of this particular study. This paper is open access paper. It is available freely. Please go through this study, learn much more because this. The next thing, the next study also by King et al. They have done what they have produced. They have produced also a renal proximal tubule with the by bioprinting technique with endothelial cells, fibroblast, and endothelial cells, epithelial, fibroblast, and epithelial cells, in vitro proximal tubule interface, interstitial interface. So what they have done, they bioprinted, they produced these endothelial cells, printed epithelial cells, and the thing, and fibroblast and epithelial cells were used for this interstitial, to develop this interstitial tissue. Now, when this bioprinted construct formed a microvascular network supported by the cellular issue deposition, then they could this interstitial space where a tight junction formed between in the 3D proximal tubule tissues and had the expression of renal efflux and uptake transporters. So this kind of anatomical structures are that is important for the function of that particular tissue. Those have also been again seen on this thing, and they have also demonstrated a fibrotic response they could find when TGF beta. TGF beta is a growth, it typically is a growth factor, but it induces fibrosis for certain tissue. Like in kidney, if there is a high amount of TGF beta expression is there, then it can lead to kidney fibrosis, fibrotic kidney. So that kind of thing also they have seen within their model. So this is, they are trying to see the physiological function of that particular 3D printed proximal tubule structures thing, because this, when they injected this TGF beta, it, the cells that is present in this, in the particular, in the tubule, that takes they express high matrix. So they high matrix deposition, they synthesize more matrix, and that getting deposited in the structure. So that because of that, the fibrosis has been generated. But when this treated treated with a cisplatin, because cisplatin is another nephrotoxic drug, what they have treated with cisplatin, they found a biomimetic reduction of viability. So the cellular viability reduced, and that is also in a dose dependent manner. And the cisplatin is a nephrotoxic drug. It disrupts the nephron structure on the cells all die, and that kind of thing also effect also they have seen. But again, when another drug called cimetidine was used, it reversed this effect, because cimetidine blocks this cisplatin activity. That present, because of this cimetidine acts on the OC2 transporter, and it, because of this, the cisplatin induced nephrotoxicity can be blocked. So this cimetidine, effect of cimetidine on the particular tissue, it confirmed the presence of OC2 transporter in those cells present in that tubular epithelial, in the proximal tubule. So this is also another study where 3D bioprinting was used to develop a proximal tubule interstitial interface by 3D bioprinting technique. It has this schematic, shows you the how this RPTEC, the renal proximal tubule epithelial cells, were printed on top of this, and the interstitial layer was printed where the endothelial cells are there, and they create this kind of vascular structures right within the interstitial space layer. And this tissue is very important for this reabsorption other thing from the proximal tubule. Right? So this is the schematic of their work, and then they have also shown the macroscopic. This is the macroscopic view of the 3D PPT tissue that is positioned on a transwell and a standard to 24L plate. Right? In the standard 24L plate, they have printed this kind of structure where this interstitial layer, and on top of that, the RPTEC monolayer is present. Right? Yeah. So then they have also characterized this by various ways. So one such thing is by histology also. They have shown this. You see in the histology, each is standing clearly showing the cellular tree and tissue organization in the interstitial. Epithelial cell. You see, epithelial is a fine layer. A tight layer of epithelial tissue is formed very clearly. That epithelial tissue is present, and here also the vascular and the other tissue can be clearly visible within this in the histological site. And then B is there is Gomor is trichome stent showing the deposition of collagen throughout the tissue. So the CD31 is basically for the vascular tissue, and the other everywhere else is the trichome stain that shows you the here the trichome stain, that is the blue color thing, that is the deposition of collagen deposition everywhere throughout the tissue by the cells present in that area in this particular tissue. C is the interstitial layer demonstrate the extensive endothelial cell line network. So these are the endothelial cell line network. So that is creating the vascular network within this interstitial tissue. So that is why with the CD31 stain it is can. And then the other thing is also RPTEC from monolayer express cytokeratin, that is red. This keratin 18. So here the epithelial tissue, only that here where the epithelial tissue that form a monolayer and they express also cytokeratin. So that is a typical marker for the epithelial cells, and you can clearly see here a tight epithelial layer is formed here in this case. And also in case of E, a collagen 4 rich basement membrane can also be seen between the epithelial and the interstitial tissue. That is very much important for, because basement membrane is the component that separates the epithelial tissue and the interstitial tissue and acts as a barrier between these two, and also it helps in filtration and other process, not filtration, absorption and other process. So there is a ECAD. So here if you clearly see the collagen 4 here present in between this tissue, and also E-cadherin, that is also a marker for cell-cell junction, that is also can be clearly seen in case of epithelial tissue. A tight junction is established here. Right? And then other thing is sodium potassium ATPase, that is also can be very clearly see in the epithelial layer. That helps in reabsorption certain solutes and all these things for the kidney further, that is present in the proximal tubule for the function of that particular tissue. So in this particular work also they have tried to reproduce a proximal tubules and interstitial interface, and also they have seen this, that they have tried to see whether this kind of tissue can, if 3D permeable tissue physiological function can be re-established or not, that kind of things they have seen. And also they have, as I earlier slide I mentioned, they use certain nephrotoxic drug to find out whether the response can be in a, that can be seen, suppose that can be seen in the human body, similar responses can be seen on a bioprinted model or not. So this is another important study where they have shown how 3D bioprinting, how a functional proximal tubule interstitial tissue interface can be established. Then there are many more studies actually they have been targeted to find out certain aspects of this or to recreate some part of this kidney structures. Like another study where by Singh et al. They published in Biometrics, where they have shown a 3D bioprinter renal tubular tissue analogs. What they have used a coaxial bioprinting, and coaxial bioprinting method we have also discussed earlier, where it has a core shell kind of extrusion nozzle, where through the core we can feed one type of material through the outer nozzle, we can outer compartment we can feed another material. And in this case they have used a crosslinker to pass through the core channel, and or probably a sacrificial material to pass through the core channel, and this outer channel a ECM mimicking biomimetic material. Like they have used kidney matrix, dissociated kidney matrix ink. So they have dissociated the kidney and thrown out all the cells, and then collected that matrix, and then they collected the matrix, and then hydrogel was developed from that, and hydrogel, and that from the hydrogel again, when you incorporate cells like this tubular tissue specific cell like the epithelial cells and other type of cells, and then you can develop this specific bioink, and that bioink was used to generate this kind of a hollow microfluidic tubes they have generated for engineering this renal parenchyma composed of endothelial cells and renal tubular epithelial cells. So these two cell types they have used to generate this kind of renal parenchyma, And then but there the function of this thing was improved by a, because they have used a functional hybrid biobank where they've used kidney specific, tissue specific, kidney tissue specific disulfide matrix along with other material to improve the microenvironment, because kidney tissue, disulfide tissue has certain bioactive factors cues that induce or that promote the function of these cells. So that because, and they also because they provide a natural mimicking biochemical biophysical environment for this thing, because and then also that helps to allow for the vascularization process in this case. Their method yielded both functional renal proximal tubule on a chip as well as implantable constructs. So basically they have shown both tubule on a chip and also implantable construct. They have both, they have fabricated with the thing, and then when they have incorporated that thing, they have transplanted this renal scapular transplantation, this sorry upon renal subcapsular transplantation of this hollow tubes that demonstrated long-term graft survival in vivo with therapeutic capability in a renal disease model. Actually, they have created a renal disease model, and there they have incorporated or implanted this kind of this tubule structures that actually helps in means they have showed a therapeutic capability of this long-term graft survival was seen. So combination with bioactive materials along with microfibrillation of this renal tubule can be produced functional kidney tissues. So this kind of work has shown the shown the promise of developing functional kidney tissue units by combining bioactive materials and 3D bioprinting with using 3D microfabrication techniques like 3D bioprinting. A kidney space, this kind of kidney specialized kidney structures, kidney tissue structures can be very well developed. But the future direction of all these works, because till now we have not achieved this bioprinting of fully functional kidney tissue for clinical transplants. We have not reached this stage. It is still there are few more further development is required in this space. Like scaling up is a major challenge for all these things, because these most of the works are like either in a microfluidic chip or in a small scale, in a micro scale. To scale up that and to produce the whole kidney is a humongous effort, is a humongous challenge. So so scaling up of those things, those functional units into fully functional kidney is the major challenge. But these can be used as a building blocks by using the concept of mini tissues. I have, as we have discussed in our earlier tissue, probably if we can develop these building blocks and if we can connect them together, then definitely we can come up with a fully functional kidney tissue. So slow, but we are have not reached there, but slowly we are progressing or moving in that particular direction. Other thing is also we need to integrate the large scale vasculature, because without that this kind of this kidney patches or this thing will not be viable or functional. So that's why we need to include the vasculature also within this thing. But ultimately the goal is to bioprint a fully functional kidney structures, and that can be used for clinical translation so that it can save many patients who are actually waiting for transplants. So please go through I have given links for the several literatures. So please go through the literature. You will understand much more about how this 3D bioprinting can be used for to develop a different kidney tissue structures, and also how bioprinting strategy can be developed further to generate full functional kidney tissue. Thank you very much.